Hello, everyone, and welcome back to this series of technical tips. I am Dr. Monica Tyagi, application scientist at Biotechnic, and today I'll be talking about the antibody drug conjugate analysis on our Maurice platform. For today's tech tip video, I will briefly introduce ADCs, explain why we need to analyze them in capillary electrophoresis, and I'm going to share some interesting data from our recently published app notes and peer-reviewed publications. What are ADCs? Let's talk about the basic structural component of an ADC. An antibody, which is typically an IgG, chosen for its high affinity and specificity. Modifications on lysine or cysteine residues define the drug-to-antibody ratio, which in short is DAR. The payload. Usually, a highly potent cytotoxin is a payload, and the number of the payloads per antibody, that is the DAR, it affects the efficacy and the cytokinetics. Another characteristic which is key for an ADC structure is a linker. It could be either a cleavable linker, which is, say, for example, an acidic sensitive or a protease cleavable and allows the drug release within the target cells. Or it could be a non-cleavable linker where the payload remains attached and is released only through the antibody degradation. ADC combine the specificity of antibodies with the efficacy of small molecule drugs, enabling selective delivery to cancer cells and reducing systemic toxicity. Why use capillary electrophoresis for ADC analysis? ADCs, heterogeneity and complexity require high-resolution analytical methods for characterization. Now, capillary electrophoresis, it's a powerful analytical technique which is widely used for analyzing antibody drug conjugates due to its high resolution and sensitivity, as CE provides excellent resolution and sensitivity in detecting differences in charge and size. ADCs often exist as a heterogeneous mixture, so CE can identify and quantify the distribution of conjugated and unconjugated species in the sample. For charge variant analysis, ADCs often possess diverse charge isoforms due to conjugation reaction or post-translational modification, impacting their efficacy and stability. CE, particularly the capillary isoelectric focusing, efficiently separate these charge variants based on charge. For aggregation and purity assessment, CE-SDS is capable of separating monomeric and aggregated ADC species, providing critical insights into purity and structural integrity. Minimum sample consumption. CE requires small sample volumes, preserving valuable ADC material, especially critical during early stage development when material availability is limited. Speed and automation. CE offers quick runtime and making it suitable for high-throughput ADC characterization in research and production settings. Here, I would like to introduce our Maurice platform. Everything that ICE does, plus significantly simpler workflow, integrated auto sampler, which gives small footprint, native fluorescence detection in CIEF mode, which gives higher sensitivity, CE-SDS mode, and an integrated analysis software, which is Compass for ICE with 21 CFR 11 compliance tool for GMP and QC environment. And if you take a quick look at the image on the left, it highlights the soft touch switch for opening the door, and the image on the right gives an interior view showing the slot where you can load the cartridges. The bottom tray is for your sample plate and the bulk reagents and buffer. You can easily swipe the cartridges based on your application needed, as the system cleans up the cartridge after the batch ends, so no cleanup is needed manually and there is no downtime. I'd like to talk about how easy the workflow is when you're working on Maurice and switching between different applications, be it icIEF or CE-SDS. Now, based on your application need, you can insert the cartridge, load the required reagents and the samples. Once you have loaded your samples, your reagents, your cartridge, you set up your batch on the software and then just hit start. You can do the method development on the go or you can have the data result once the run is completed. Now let's look into ADC analysis on Maurice platform. As you all know now, an important factor concerning the efficacy, pharmacokinetics, and stability of ADC is the drug to antibody ratio, just DAR, and which is defined as the number of drug molecules conjugated to the antibody. Attaching too many drugs to the antibody can lead to low efficacy, and attaching too many drugs can lead to high clearance rates, instability, and systemic toxicity. Therefore, the DAR must be carefully monitored during the synthesis of all ADCs, and measures should be taken to ensure the DAR homogeneity. Now, the lysine-linked drug conjugates, for example, have a charged isoforms that are more acidic, and icIEF is a useful method for assessing the amount of unconjugated MAB relative to the conjugated MAB and for providing a rapid way to monitor conjugation-related changes. However, certain limitations still exist, as lysine linker without a conjugated drug also shift charged isoforms to the acidic end, leading to possible overestimation of the conjugated MAB in the sample. To address this concern, the Maurice system introduced fluorescence detection at 458 nanometer in addition to the existing absorbance and native-induced fluorescence detection mode. Now, the new fluorescence 458 nanometer filter is particularly useful for ADC characterization because many ADCs absorb at 280 nanometer and have fluorescence in this region, allowing for the quantification of the conjugated drug independently of the antibody for each charge isoforms. To dive into more further and have more details, please refer to our application note on the ADC filter. Here and in few upcoming slides, I will be discussing our recently published application note on ADC analysis on Maurice platform. This app note highlights analysis of ADC that have pH labile linkers, onboard mixing capability of Maurice, the 458 nanometer fluorescent filter that I spoke about earlier, low fragmentation and high throughput of ADC analysis using Maurice CE-SDS. This study was conducted on two trastuzumab ADCs, Kadcyla, having a non-cleavable thioether linker, and Enhertu, which uses a cleavable tetrapeptide linker to conjugate topoisomerase 1 inhibitor. Now, these differences in conjugations among ADC types, they significantly influence their chemistry and stability, and which can be accurately monitored by capillary electrophoresis. For the size variant analysis using CE-SDS application, ADC samples were stressed at 60 degrees Celsius for 1 day and compared to the unstressed material, with the default separation conditions for the reduced samples and a 5-minute separation time extension for non-reduced samples on CE-SDS Plus and a 2-minute on the Turbo CE-SDS cartridge. This figure shows the CE-SDS analysis of Herceptin, trastuzumab, and two ADCs, Kadcyla and Enhertu, which both are trastuzumab-based ADCs, under non-reduced conditions, as you can see in figure A, and reduced conditions in figure B, and the analysis of Enhertu under non-reduced and reduced conditions in figure C on Maurice platform. The only notable differences between the two Enhertu profile was the presence of two high molecular weight species detected under non-reduced conditions that align with the heavy light, heavy, and the heavy light variants of Herceptin. Now, the high degree of comparability in the CE-SDS profile of Enhertu under reduced and non-reduced condition is likely due to its site-specific conjugations where drug linker molecules are attached to the hinge region cysteine groups. Now, the 4 hinge region disulfide bonds are replaced with the 8 drug conjugation sites, and this new intact structure is not covalently bonded like the original antibody. Though Enhertu is highly homogeneous with an average of DAR of 8, some literatures report a DAR of 7.7 indicating remaining disulfide variants. And this offers a plausible explanation for the Herceptin and Kadcyla. The intact peak is seen under non-reduced conditions along with other minor peak where the light chain, the heavy chain, and the non-glycosylated heavy chains are detected under reduced conditions. In contrast, Enhertu has a similar profile under both non-reduced and reduced conditions for the comparable migration behavior observed in both CE-SDS mode, but with 3% more higher molecular weight species for non-reduced CE-SDS analysis. Additionally, to have the same great data with faster speed, we did comparable study on our CE-SDS Plus and Turbo cartridge. On the left, you can see the image of both of our cartridges, and on the right, the comparable high-quality data on Enhertu with 4x faster speed on Turbo cartridge. We showcase the data comparison of all three candidate molecules for percent purity analysis using CE-SDS Plus and Turbo cartridge with 4 times faster on Turbo cartridge. You get the data you can trust at the speed you need. Now, moving on to the charge variant analysis using the icIEF applications of Maurice. In addition to being the gold standard for charge heterogeneity analysis, icIEF has long been known to be a stability indicating method for a variety of molecules. Now, here for this application note and for the molecules we were studying, this is our platform method for MAB. And for analyzing the ADCs, the modifications were made to switch the PI marker because some ADCs have 458 fluorescence. Now, there are a couple of different fluorophores on these PI marker, but these ones work out best and really nicely because they are fairly equal in both the chat. This figure demonstrates the power of icIEF by comparing the profiles of native samples with the stress sample for each of the three molecules, Herceptin, which is you can see in the figure A, Kadcyla, figure B, and Enhertu in figure C. Marked differences between the charge heterogeneities of native and stress samples are observed, with degradation confirmed after the samples were incubated at 60 degrees Celsius for 1 day. An increase in acidic species is observed in all three degraded molecules. The another key feature is the use of onboard mixing on the Maurice system. It significantly enhances the analytical workflow of ADCs by automating the sample preparation, ensuring consistent reagent distribution, and reducing variability in the critical assays. By streamlining the sample preparation, minimizing the sample loss, and conserving the valuable material, onboard mixing ensures precision and data integrity, meeting the high regulatory standards required for ADC development and characterization. Now, in particular, for a pH-labile linkers, where the pH-mediated ring opening of succinimide linkers occur, when acid or the base conditions, they hydrolyze the cyclic structure, as you can see in the figure on your right, that leads to the formation of an open-chain derivative. Now, in antibody drug conjugate, this reaction can influence the stability and drug release profile by altering the linker drug architecture. This process is important in determining the ADC behavior, since the linker stability at different pH level ensures precise drug delivery and minimal off-target effect. In this current study, this approach was chosen to minimize sample handling and prevent degradation of Enhertu, which is a pH-labile ADC. In this study, OBM was leveraged for the analysis of all three molecules. The most notable differences were observed for Enhertu. As you can see in figure A and B, it shows a comparison of Enhertu analyzed with the standard icIEF method and the Maurice OBM method respectively. In turn, each figure shows charge profile of a sample resulting from the analysis over a batch of 49 injections. The later injections in the batch are prone to pH-induced degradation with the standard icIEF method, as shown in figure A, while they remain relatively stable when analyzed with the Maurice OBM method, as shown in figure B. I would like to share one of the recent peer-reviewed publication on ADC analysis using the icIEF application on Maurice platform. This work encompasses the development and optimization of icIEF method for high DAR ADCs of various DAR values ranging from 4 to 8 and a payload linker chemistry. The method optimization here focuses on improving the resolution and stability indicating capabilities and differentiating contributions from the protein and payload linker. For more details and further reading, please do scan the QR code to read this article. And I'll be sharing a couple of interesting data in few slides from this article. In this article, the author emphasized on the solution stability of the icIEF method, and it must be evaluated considering the possible instability of the payload and or the linker. Now, the figure on the left shows the electroferrogram for the map. The icIEF separation was successful and achieved using a 3 molar urea and 2% formaldehyde 3 to 10 in a sample matrix without using a narrow range amphylite. Now, figure on the top right shows the E-gram for the ADC, where you can see the multiple peaks were observed with similar intensity at the initial time point, with the PI ranging from 7.6 to 8.1, while the PI of all the peak range from 7.3 to 8.5. Now, the peak pattern is different from the map, as you can see, which is on the left. Now, due to the high DAR, which could have the potential to impact the charge exposure during the separation, a narrow amphylite, which is 8 to 10.5, in addition to the wide range formaldehyde, was used with the attempt to achieve an acceptable separation. The multiple peaks were observed with similar intensity at the initial time point, with the PI ranging from 7.6 to 8.1, while the PI of all peaks ranged from 7.3 to 8.5. Now, again, the opening of the lactone ring generates an additional negative charge during the sample preparation, causing the method unable to capture the original charge state of the ADC with a poor method robustness. Considering the optimal stability of the lactone is under weak acidic environment, the alkaline formaldehyde 8 to 10.5 was removed while the formulation buffer was used as the matrix. Now, the solution stability here is significantly improved under these conditions, and as seen in the E-gram on the bottom right. Under the optimized conditions, there were no interfering peaks in the formulation buffer. The stretched sample showed increased basic variant and with well-defined separation of both acidic and basic peak compared to the unstretched sample. The method was able to differentiate the map from the ADC once it was optimized on the Maurice system. Another interesting article that I would like to highlight is the one that where the author have used the CE-SDS applications to analyze the ADCs and showcase CE-SDS as on Maurice as one of the powerful tool for studying these molecules. For more details, please scan the QR code for the article. For more resources on ADC analysis on our Maurice platform, please scan the QR code. And thank you for listening.